ऑनलाइन क्लास हवा में वो घर থেকে পড়িও
So today's topic for discussion is origin of eukaryotic cell.
यस सर
All we know that what is eukaryotic cell. Yesterday's class also. Kindly switch off your mic. Okay, so what is eukaryotic cell? Yesterday we have discussed the cells which have described or properly described nuclear membrane. That means nuclear space is well defined. That is known as the eukaryotic cell. But in prokaryotic cell, the nuclear part, the nuclear region is not well defined. That is the basic difference between these two. Other differences between prokaryotes and eukaryotes, they include what? They include, firstly, prokaryotic cells. They generally do not have all the membrane bound cell organelle. But in case of eukaryotic cells, they have membrane bound cell organelles. Prokaryotic cells, in on the basis of their packaging of genetic material, they are also different from eukaryotic cell. Cytoplasmic organelles, already I have discussed about cytoplasmic organelles, that is the membrane bound organelles. Cell wall composition. In case of eukaryotic cell, the cell wall composition and prokaryotic cell wall composition is also differ from each other. Nuclear region is also different because in prokaryotic cell there is no proper nuclear region is found, but in eukaryotic cell nuclear region is distinct and well defined. Nucleus is the largest and distinctly visible organelle in the cell. Generally, prokaryotic cell do not use microtubule at the time of cell division, but eukaryotic cell generally use microtubules at the time of cell division. And also, cell size is different in when we are comparing prokaryotic cell and eukaryotic cell, both have different cell structure and size. Okay, so these are the basic differences between prokaryotic cell and eukaryotic cell. Prokaryotic cell origin, little bit we have discussed in previous class. Sir, sir, मैं detail में इग्नोर क्लास कंटिन्यू करूँ चलते हैं हम क्लास सारी की कॉल कर कोई वेट कर बेसिक साइंस का है सर 5:30 से 1:30 क्लास होची मॉर्निंग से थोड़ा कॉल कर आगे हाँ ठीक আছে সেই ইগনোরে হবে তো আগে হ্যাঁ ঠিক আছে স্যার ଆଜ୍ଞା ନାଇଁ ସାର୍ ନାଇଁ ଏଇଟା ନୁହଁ ସାର୍ ମୁଁ ଆପଣଙ୍କୁ ଏଇଟା ମେସେଜ କରିଦେବି ହ୍ୱାଟ୍ସଆପ ନାଇଁ ନାଇଁ ସାର୍ 82800301 Oh, WhatsApp number. 82803901. हाँ आगे आगे सर ठीक ठीक है आगे आगे सर okay sorry for the interruption it's a call from igno okay so we are going to start about the origin of eukaryotic cell before just and a brief we are going to discuss about the origin of life because eukaryotic cells are the modified cells that means the primitive cells are prokaryotic cells the simplest form of cells are prokaryotic cells and the complex cells are the eukaryotic cells am I audible to all
Yes, sir.
okay so yesterday day before yesterday we have discussed in detail what is prokaryotic cell, how they originated, Popper-Halden concept, Miller-Urey experiment, everything. That pre-atmospheric condition, how becomes suitable for life, how chemical synthesis occurs. That means chemogeny occurs first, then that lead to biogeny, formation of biological substances, biological entities. Okay, so chemogeny followed by biogeny. These are the basic idea for origin of life on Earth. Then the biological substances, when acquired, certain nucleic acid material like RNA, DNA, all this kind of nucleic acid material, they converted to complete cell. Okay, the primitive cell. So this was the basic process of origin of life, what were described by the scientist, previously described by the scientist. Okay. So in next slide, these are the basic features already we have discussed how Miller-Urey experiment explained about the pre-atmospheric condition, about the reducing atmosphere, about the oxidizing atmosphere, everything. Now, the origin of eukaryotic cell. The leading theory of origin of eukaryotic cell evolved from a fusion between archaean cell and aerobic bacterium, a process called eukaryogenesis. The process is called as eukaryogenesis. That means how symbiotic relationship developed between these two, between archaea and bacteria. That helps in the formation of what eukaryotic cell. Yesterday also in brief we discussed about the endosymbiotic theory. Endosymbiotic theory explains us in detail how eukaryotic cell originated. So this is the pictorial presentation of eukaryogenesis. How eukaryotic cell evolved. How eukaryotic organisms they developed from different concepts. So this theory, endosymbiotic theory, what it explains, endosymbiotic theory, life actually began with prokaryotic cell. Okay, that is the bacterial cell and archaeal cell, which was about 3 to 4 billion years ago. Eukaryotic cells just they began or estimated to begin before 1.6 to 2 billion years ago. Okay, so for that, certain theories are very important. That is, the first theory is explained that endosymbiotic theory. This theory explains us what? This theory explains eukaryotic cells evolved through a symbiotic relationship between different prokaryotic species. As I have already mentioned, the prokaryotic archaea and bacteria, they develop a symbiotic relationship between them to generate another kind of cell. So ancestral archaean, generally archaea is considered as the host cell. Okay, that host cell engulfed a bacterium that might by chance or by plan. Okay, whatever may be the cause, but ultimate result is the host cell, the archaea cell, the larger cell, they engulf the smaller one, the bacterium. Okay, which capable of, so which kind of bacteria, normally the larger cell when ingested, the small cell, general small cell, simple small cell, that doesn't have any meaning because simple small cell is just a food for the larger cell for certain time. They do not develop symbiotic relationship because in a later extent, the small one become parasite on the large one. Large one will not get any kind of help from the small one. That's why they simply ingest and digest the small cells after some time. But when by chance they engulf bacteria which are capable of aerobic respiration, that means the small cell which is capable of aerobic respiration, aerobic bacteria, that means in presence of oxygen, they can produce the energy in the form of ATP. Then only the cell, without engulfing those, they can easily obtain their energy. So it's a benefit for the larger cell. This is the nature of all the entity that if we are getting some benefit, then generally we do not cause harm to that species or cell. When the larger cell is getting benefit from the smaller one in the form of energy production, the process of energy production, then the small one is protected by the larger one, not harmed by the larger one. That is essential. Okay, that is the endosymbiotic theory. That means they engulfed the small one and they made a symbiotic relationship. Not only the small cells got benefited from this process, also the large cell getting the benefit from this process. Okay, for that, instead of digesting it, they formed a mutualistic relationship. Mutualism. That means you help me, on that basis, I'm going to help you. Okay, that is the mutual relationship between these two. So the engulfed bacterium eventually evolved into mitochondria. It was hypothesized that the engulfed bacterium eventually they converted to mitochondria. Okay, because the function of mitochondria is almost similar to the function of ancient aerobic bacterium. Okay, that's why endosymbiotic theory is widely accepted theory, worldwide accepted theory for origin of eukaryotic cell. And later, in plants and algae, in the same manner, a eukaryotic cell engulfed a photosynthetic cyanobacterium in the place of aerobic respiratory bacteria. Then that photosynthetic cyanobacterium is converted to, in later phase, chloroplast. Okay, so these are the basic explanation of endosymbiotic theory? I think all of you clear about the endosymbiotic theory.
Yes, sir.
Yes, sir.
Okay. So this is one theory explained by different scientists,
Yes, sir.
endosymbiotic theory. Here it is mentioned that the engulfed bacterium allowed to archaean cell to use oxygen to release energy from nutrients and to protect the bacterium from predators. Over time, the two organisms developed a symbiotic relationship, and the descendants of the engulfed cell are today's mitochondria. In the same manner, chloroplast also evolved. So we can take some evidences which are in support of endosymbiosis. That evidence means which are the proofs. Like mitochondria and chloroplast have their own DNA. Though they are present inside the cell, they are considered as cell organelles. In some certain manner, they are considered as cell organelles, but still they have their own DNA, their own genetic material. They also separate from nuclear DNA. Their DNA is different from nuclear DNA, though they have the same cell structure. And it's circular like bacteria DNA. The mitochondrial DNA, the chloroplast DNA generally circular bacterial DNA. Okay. They have double membranes like the ancient cell, the prokaryotic cells. They replicate independently by a process similar to bacterial fission. Their replication is also similar to asexual reproductive mode found in case of bacteria. And their ribosomes are 70S type, 70 Sperbox unit. That is bacterial type, not like 80S, which is found generally in eukaryotic ribosome. So these evidences support the theory of endosymbiosis. Okay, so these are certain evidences. Then the next theory, the inside out model. The inside out model. So this inside out model explains us what? The model suggests that eukaryotes evolved from a prokaryotic cell with a single membrane that extended protrusion which focus on the cytoplasm and endomembrane system. Okay, so what is the explanation for this inside out model? It is proposed by Baum and Baum. What this inside out model explains, actually, its explanatory idea, just remember this. Just listen this carefully, that instead of a host cell engulf, means whatever written here, I'm just explaining these points. This inside out model, whatever written here, I'm just going to explain in detail. So instead of host cell engulfing another cell, which was in classic endosymbiosis model we have discussed, the host cell sent out protrusion. Protrusion means simply the extension. Okay, extension of what? Extension of its membrane to surround and trap bacteria that live on its surface. Okay, inside out model explains. Again, I am saying they extends protrusion. Protrusion means what? Here it is mentioned that single membrane that extend protrusion. Protrusion means the extension of the membrane. Protrusion to what? Towards the surround. If at this surround the bacteria is present, they fuse, they trap the bacteria which lived on its surface. If due to any chance factor, if the bacteria come in contact to that surface, they just develop the protrusion and trap the bacteria. This is the case of inside out model proposed by Baum, Baum, Baum. So for this, for the analysis, we can explain certain steps that an ancestral archaea or archaeons from blades or arms. Okay. In this structure, just a minute. If this is the cell and on the surface a protrusion or any kind of outgrowth occurs, that is generally known as blebs. Actually, the pure bleb structure is not in this picture, but bleb concept is that. Okay, if any outgrowth or development occurs, that is known as the blebs. So these arms, these outgrowths, they wrapped around symbiotic bacteria, specifically the alpha proteobacteria ancestor, which was considered as the ancestor of mitochondria, the aerobic bacteria named as alpha proteobacteria that lived on the archaean surface. Okay. Over time, arms fuse together, trapping the bacteria inside and forming the early cytoplasm. That's why this is called as inside out model. So outer structure, they just trap the bacteria inside. The space between the original archaeal cell and the outer membrane become the cytoplasm. Okay, this is the step. And the internalization of these symbionts eventually led to formation of mitochondria. When they ingested into the cell due to any manner, due to endosymbiotic manner, due to inside out manner, when they ingested inside the cell, they become the part of the cell and their function become the part of the cell's function. If they are producing ATP, they are producing energy in form of ATP, that is also the cell's function. We can say that the cell has the capability of production of ATP. Got it? So this is all about the inside out model. Okay. Now, the diversity of eukaryotes, it includes microscopic single cell organisms as well as animals, plants, and fungi. Okay. So single cell eukaryotes, it generally have nucleus. They have membrane-bound organelles like mitochondria, Golgi apparatus, everything, but they are single cell. They are not multi-cell. Okay. Certain examples like amoeba, paramecium. Okay, so these are certain examples of these cells. So there is diversity. Various kind of eukaryotic cells are there. Single cell eukaryotic cells are there. Complex, simple organisms, having eukaryotic cell, complex organisms having eukaryotic cells. So these are different types of eukaryotic cells. That is a great variation in eukaryotes. Now come to the characteristic of eukaryotic cell. Many of you well known to these characteristics. They have well-defined nucleus. Well-defined nucleus, and the nucleus is true nucleus. That's why it is named as eukaryon. Karyon means nucleus, eu means true. The DNA is enclosed inside nuclear membrane. The membrane is well defined. The membrane, nuclear membrane is well defined. That means the nuclear space is well defined. Okay. Then membrane bound organelles like mitochondria, organelles like chloroplast in plant cell, Golgi apparatus, endoplasmic reticulum, all these kind of organelles are present in eukaryotic cells. Another important peculiar property of this eukaryotic cell, they have linear DNA. That means DNA is not circular, it is linear. Okay. And organized into chromosomes. The linear DNA are organized into chromosomes. The complex structure, that means the cells have compartmentalization. Compartmentalization means what? Different organelles perform specific function. These are very important terms you have to remember. Compartmentalization is found in case of eukaryotic cell, which is not present in case of prokaryotic cell. Compartmentalization, again I am saying the meaning of compartmentalization is the different cell organelles are present for different function. But in case of eukaryotic cell, as they are very simple cell, they have very less organelles, and all the functions performed by very less organelles. Okay. Presence of cytoskeleton is another important property. Internal network of microtubules, microfilaments, they are made up of proteins. Microtubules, microfilaments, intermediate filaments support the transport of intracellular components and help maintain cell shape. They act as skeletons, okay, to maintain the cell shape, for the process of transportation, everything, they are active. Then advanced cytoskeleton also, these are known as advanced cytoskeletons, which are meant for safe movement and division. Sexual reproduction is found in case of eukaryotic cell. That means mode of microtubules, meiosis. The eukaryotic cell undergo meiosis. They undergo the process of fertilization. Fertilization means involving gametes and genetic recombination. Mitosis for cell division also it found, which ensure equal distribution of chromosomes. Eukaryotic cells have multiple chromosomes, rod-shaped chromosomes, where most prokaryotic cells have a single circular shaped chromosome because DNA is circular. Okay. Different diverse form. Already we have discussed in the diversity that unicellular and multicellular category of eukaryotic cell. The cell wall is another important property of this eukaryotic cell, which is not present in all eukaryotic cells, but it is present in certain eukaryotic cells. Cells like plants, they have cell wall, but this cell wall made up of cellulose. Okay, this cell wall made up of cellulose, which is different from the prokaryotic cell wall. Flagella and cilia, they also present if they have flagella or cilia, these structures have 9+2 arrangement of microtubules. That means microtubules arranged in that manner, 9+2 manner. Okay. So these are different important property of eukaryotic cell. Along with that, locomotion is another important property of this eukaryotic cell. Already we have discussed about the flagella and cilia. They are the locomotory organs. This locomotory organ helps in the movement of eukaryotic cells. This is the structural view of eukaryotic cell. These are different organelles. Just look at here. Eukaryon. Why we are emphasizing the karyon, the nucleus? Because this is the largest organelle of the eukaryotic cell. It is the distinct organelle of the eukaryotic cell. They have endoplasmic reticulum, which is the continuation from, generally it is considered as the continuation from plasma membrane to the nuclear membrane. Okay, Golgi complex is there, mitochondria, lysosome, ribosome, plus plasma membrane, vacuole, microtubules, everything is present in general prokaryotic cells. I think all of you clear about this. Up to this, if any confusion, you can ask me.
No, sir.
No, sir.
Then come to the first eukaryotic cell, which was considered as the first eukaryotic cell, and how, when, where it derived or originated. The earliest eukaryotic fossils, they found around 1.5 to 1.8 billion years ago. Okay. Acrid touch is a microscopic organic structure throughout to be eukaryotic microfossil. Microfossil, that means the fossil size less than 1 micron or 1 micrometer. That fossil is known as microfossil. So these archetypes are considered as the fossil or considered as the first eukaryotic cell. They present some of the earliest eukaryotic life form. Okay, they a larger and more complex eukaryotic fossil appeared after 1.5 billion years ago. Means before that, whatever fossils were found, they were smaller eukaryotic cells at their origin. But before 1.5 recent time, the origin of eukaryotic cells occurs. Gryphenia spiralis is another name of this eukaryotic fossil. They are found approximately 1.6 billion years ago. Is one of the earliest known fossil of eukaryotes, thought to be a multicellular alga. Okay, so these are certain pictures of this eukaryotic fossil. Then diversification of eukaryotes. Just look at here, certain tree structure. In yesterday's class, we have discussed about the tree structure, how phylogenetic trees were formed, what is the manner of tree formation, and on which basis we can form trees, phylogenetic trees. These are very important aspects. Okay. So diversification of eukaryotes, following their origin, eukaryotes diversified into several lineages, including plants, animals, fungi, and various protists. Early eukaryotic diversification was driven by acquisition of new metabolic pathway. In You might remember in yesterday's class, I have discussed that the eukaryotic cells, they originated from the prokaryotic cells. They originated from the prokaryotic cells. The diversification point always focus on the process of mutation, and the mutation is caused by any kind of environmental change, environmental pressure. Okay, so this is the process of diversification. So how this diversification of eukaryotes occurs, that is very important. After origin of eukaryotic cells diversified into many forms. This is, just look at this picture, the origin point, then eukaryotic, different form of eukaryotes, they diversified to different groups. Okay, this is the main origin. From this main origin, or we can say it as LUCA, last universal common ancestor, from this eubacteria, archaebacteria, eukarya, everything divided. This eukarya, again, they develop more stability due to more adaptability to the environment, changing environmental situation. Because most of the eukaryotic cell, they focus on sexual reproduction. And we all know that sexual reproduction is the important concept of variation. If variation is there, then only we can make adaptation to the environment. Because if environment is static and organisms show variability, that will cause harm to the organism. But our environment in which we are residing, the environment is not static, it is dynamic environment. So in dynamic environment, if organism becomes static, then it is difficult for the organism to survive. That's why organism has to explain multi-dimensional adaptability to accommodate itself with the changing environment. Otherwise, it will be difficult for the organism to survive. Okay, so this is the major diversification of eukaryotic cells. The diversified plants, they diversify to animals, fungus, various protists. Major events for the diversification, some events we have discussed. Endosymbiosis, one of the major events for the diversification. Sexual reproduction is another event for the diversification. Multicellularity is the event for diversification. Colonization is another event for diversification. Okay, so these are different events for diversification of eukaryotic cells. Major eukaryotic groups, if we discuss, the Opisthoconta. Opisthoconta means animals, fungi, Archaeplastida. They belong to plants, green algae, red algae. SAR group. SAR group means Stramenopiles, alveolates, Rhizarians. All these are different SAR groups. Excavata, including Euglena, Giardia, they are called as excavata. Okay. Amoeboja. Amoeboja means amoeba. Slime molds. They are under the amoeboja. Okay. So these are different groups, major groups of eukaryotes. Sorry. where at this slide is this is the diversification trees of eukaryotes. Now come to measure evolutionary transitions of these eukaryotes. What are the major evolutionary transitions? First is symbiogenesis. Symbiogenesis, origin of mitochondria and chloroplast. Already we have discussed, this is the picture, how the aerobic proteobacterium, which was considered as the ancestor for mitochondria, they engulfed by the larger cells. Okay, then they reside in the cell, develop a symbiotic relationship with the cell. So these are the transitions, major evolutionary transitions. That means the simple eukaryotic cell is getting converted to complex eukaryotic cells. Okay. Sexual reproduction is another evolutionary transitions. Multicellularity, everything we have discussed. Sexual reproduction is one of the important cause, because when there is sexual reproduction, there is recombination factor. If recombination factor occurs, then that lead to change in the structural property, because exchange of genetic material occurs. That is the reason for the variation. If genetic material exchange occurs, that leads to the variation in the species. Multicellularity also, the emergence of multicellular organism laid the foundation for the development of larger and more complex organisms. If multicellularity does not occur, then complex organisms evolution might not come to the earth surface. Okay. So this is all about the eukaryotic cells, their properties, their structural analysis, everything, and their evolution. All of you clear, I think? Any questions from this part? If any questions, then ask me. I'm trying to clear your doubts. Otherwise, we'll go to the next part. From the last unit, we left this part. I'm going to discuss about this part, bacterial genome evolution. Origin of genome by horizontal gene transfer. In yesterday's class, we have discussed about horizontal gene transfer. Okay. So by this process, the bacteria acquire genetic material from other organisms rather than inheriting it from a parent organism. Okay. Yesterday, I have explained that what is vertical gene transfer and what is horizontal gene transfer. If inheritance occurs, genetic material transfer from parent to offspring, then it is known as vertical gene transfer. If apart from parent, any other bacteria is responsible for transformation of genetic information, then it is known as okay. So what is the mechanism for this horizontal gene transfer? Three important mechanisms are there: transformation, conjugation, and transduction. This is bacterial transformation. How transformation occurs? This is the clear-cut presentation, the pictorial presentation of bacterial transformation. Is the process by which bacteria take off foreign DNA from their environment and incorporate it into their own genome. Okay, that means that genome, that DNA was not previously present on that bacteria. It was acquired by the bacteria from the surrounding environment. Surrounding environment means from the surrounding organism, which have that kind of genetic material. So basic steps already mentioned in this picture. I'm going to explain according to the picture. Just look at these things. Uptaking of DNA is the first step. Here it is mentioned that uptake DNA. Transformation is the process of DNA uptake by bacteria from the surrounding environment. The cells that have the ability to uptake are known as competent cells. Okay, this process was first reported in Streptococcus pneumoniae by Griffith experiment, Frederick Griffith's experiment. So, what is that uptake of DNA? A bacterium picks off free DNA fragment from its surrounding. That is the uptaking of DNA. Here, this is the DNA from other organism. Okay. And from this DNA, we have to select the particular portion, this portion. And we can cut this portion by using any restriction enzyme, which are considered as molecular scissor, which can cut the DNA at a particular point. Okay. Now this genome is the environmental genome. That means which is not primitively present in the bacteria. This is the competent bacteria. This bacteria is the competent bacteria. That means this bacteria has the ability to receive the DNA, uptake the DNA. Okay. Just look at this picture. This kind of, this color, this type of DNA was not primitively present in the bacteria. Okay. So second step is integration into genome. That means this foreign DNA is either degraded or integrated into the bacterial chromosome or maintained as a plasmid. Got it? That means this should be integrated into the bacterial genome. Same restriction enzyme help in cutting the genome at a particular portion. Okay. Molecular glue are used to fix this portion. Then we can see the next step, which is known as expression. That means if the DNA is integrated successfully, in this picture, the DNA is successfully integrated by using DNA ligase, molecular glue. So the DNA is integrated successfully. That means particular codon, particular sequence is allowing that DNA fragment, the genetic fragment, to integrate successfully into the host cell, inside the host cell. Then the bacterium may express new traits. Now, from the next generation, the bacterium will express the new trait. That means combination of genetic material. Just look at the previous genetic material. The color is like this. But the next genetic material The variation in color. Variation in color means there is change in genetic material. This is the concept. All of you clear, I think? Bacterial transformation
Sir,
or
I
any
have a
confusion
question.
in yes?
Sir, the foreign gene can integrated into plasmid DNA or the bacterial DNA, the nuclear DNA.
Not getting you. Again, the question.
So the foreign DNA that will be integrated into the plasmid DNA or nuclear DNA.
Means
Bacteria
during
has,
transformation process.
yes, sir. Because the bacteria has plasmid DNA and nuclear DNA. Both are present in bacteria.
Okay, foreign first think about the matter that foreign DNA have a same homologous sequence with the bacterial DNA. Yes or no?
Yes, sir.
That's why we are uh, integrating the foreign DNA. We have certain desire desire to express the DNA. That's why in general case, we should incorporate the foreign DNA into the main DNA of the bacteria. So it is the main DNA. It is what we call it as bacterial DNA. Sometimes if we need, then it's a plasmid or circular DNA, which is independent, which is replicate without integrating. We can incorporate, but generally it is incorporated in the main DNA of the bacteria. Okay.
Okay, sir.
Then the second process, the conjugation process, bacterial conjugation. Just look at this picture. First thing, bacterial conjugation involves these following steps. That is formation of pilus, also known as sex pilus. The donor bacteria, one is here when we are talking about the conjugation, one is donor, another is recipient. Okay. The donor have certain property which is not present generally in case of recipient. So bacterial conjugation explains what? Bacterial conjugation explains the direct transfer of DNA from one bacterium or bacterial donor to another bacteria. This is the concept. Then the donor cell has special DNA cells, which is generally called as F plasmid. Why F plasmid is important? It is considered as the fertility factor. Then recipient cells, which lacks the fertility factor or F plasmid, and the pilus, the hair-like structure that connects the donor and the recipient. This is important phenomenon. Okay. So these are the steps. Formation of pilus. Just look at this picture here. This is the pilus. the F positive, that means which have fertility factor. This extend cells, which is called as sex pilus, towards the F negative. F positive is the donor, F negative is the recipient. Clear? Then attachment occurs between these two. Attachment means when this develops, when the pilus is arising, they come in contact with the recipient, or the recipient come closer to the donor. The pilus attached to the recipient and retracts, or pulling the two cells together. This is a bridge. This is a conjugation bridge between these two. So it is known as what? It is known as, in this case, it is known as the mating bridge. It is considered as the process of sexual reproduction because two cells are involved. But this is so many scientists, they generally do not consider as the sexual reproduction category, but some of them they consider is at sexual reproduction category because two cells, two parents are involved in this process. Okay. So this is the mating bridge between these two. The mating bridge formed between those two cells helps in transformation of DNA. The F plasmid is caught and nicked at bacterial site, okay, which is called as origin of transfer or ET site, and a single strand of F plasmid DNA move into the recipient. Just look at this picture. Clear? So this single strand of DNA is transforming to the next cell which do not have that kind of genetic material. Then DNA replication now occurs. Both the cells synthesize the complementary strand. Now both the cells have complete F plasmid. That means the primitive one means, I mean to say that the F plus or bacteria having fertility factor, and the F minus, the bacteria do not have fertility factor. Both now become same. Same means they have fertility factor in both the case. Both bacteria become F plus cell and can conjugate with other F minus cells. Okay, so these are the process or the mechanism of bacterial conjugation. There are two things mentioned here. One is relaxome. Relaxosome is mentioned here. Just look at this. Here it is mentioned that relaxosome. What is that relaxosome? It's a protein DNA complex that form at origin of transformation on a plasmid during bacterial conjugation. I have explained the origin of transformation, where it segregated single strand structure. Okay. It prepares the plasmid DNA for transfer to the recipient cell. The main function is to bind to the ORT sequence on the plasmid, cut one strand of the plasmid at the specific site, and unwinds the DNA slightly to begin transfer. Okay. Another is there, which is known as transferosome. What is that transferosome? It's a protein complex also that forms the transport machinery during bacterial conjugation. It helps move the DNA or prepare by relaxosome from the donor cell to the recipient cell through the conjugate bridge. The main function of this transferosome is what? Is building the mating bridge between donor and recipient. They forms a channel. They help to pull the DNA into the recipient cell during transfer. So this is all about the detailed description of mechanism of bacterial conjugation. If any question, then you can ask.
Sir, could you please repeat the meaning of fertility factor?
The meaning of
Fertility
fertility
factor.
factor.
Sir.
Uh, when we are explaining this F plus or F factor or F plasmid, is a generally considered as a plasmid, which is small circular DNA. It plays a crucial role in bacterial conjugation. Okay, it's a key component that determines a bacterial ability to act as a donor. Got it? Generally, it takes place in the process of bacterial conjugation. It is commonly referred to as plasmid. Got it?
Yes, sir.
Any other questions? If not, then we'll go for the next slide. This is the third process. This is bacterial transduction. Bacterial transduction. What it explains actually, the definition of this transduction is the process of transfer of gene from the recipient to the donor through bacteriophage. Okay. So it's a specialized process. What are the steps? The diagram is in front of you. The specialized stage for this transformation is bacteriophage infection is the first phase of this transformation. Bacteriophage infection means a bacteria. This is a bacteria. The structure shows that this is a bacteria. A virus which infected by bacteria is known as bacteriophage. Okay. It infects a donor bacterium by injecting its DNA. Got it? The donor bacterium by injecting its DNA. It is the first step. The second step, the phase replication. Phase replication explains what? The phase DNA takes over the bacterial machinery, making the host bacterium produce new phase particles. Okay. So here, this is the bacteriophage. They first fix with the bacteria, then they incorporate their genetic material, which make a change inside the bacterial cell. What type of change? That means the genetic material which are not earlier present in the bacterial cell, now they develop inside the bacterial cell. Okay. Then the next phase, the third phase, is the packaging of bacterial DNA. This is the packaging process. That means during this process, sometimes the bacterial chromosome is accidentally packaged into new phase particles instead of viral DNA. Okay. Then bacterial DNA transfer, newly formed phage now carrying bacterial DNA. They infect a recipient bacterium. Again, the recipient bacterium which was not infected earlier, they are infected by bacteria. Okay, so the bacterial DNA. Then the genetic recombination is the next process in which the transfer of bacterial DNA can integrate into the genome of recipient bacterium through the process of recombination. The recombination process is seen here, which was not earlier present, but now exchange of material, genetic material, leads to the process of genetic recombination. Various kinds of bacterial transcriptions are there: general transcription, specialized transcription. Okay, so various kind of transcriptions are there. Got it? So all about the bacterial transcription. Any confusion regarding bacterial transcription? You may ask question. You can ask any questions.
Sir, please repeat once again shortly.
Not audible properly.
Please repeat it again shortly.
Means the
Yes, sir,
transcription
repeat
process
it.
is okay. So generally the phage means bacteriophage. It's a in general concept, we can say the bacteriophage is a type of virus. Okay, which specifically infect bacteria. That's why it is considered as bacteriophage. Bacteriophages are present everywhere in nature and they play a major role in regulating bacterial population. Okay, so these bacteriophages actually infect the donor bacterium by ingesting its DNA. Then the second step is phase replication. That means the phase DNA takes over the bacterial machinery. Okay, they manipulate the bacterial machinery. Bacterial machinery means the DNA replication machinery. They manipulate the bacterial DNA. They have that much of capability. They can manipulate the DNA replication machinery, making the host bacterium to produce new phase particles. Okay, just look at here in the third picture, you can see this is the manipulation, which was not earlierly present. Next is the packaging of those manipulation. Packaging of those manipulation means the bacterial genome, they have the capability to develop their envelope. That is the packaging. In simple manner, we can say. Then bacterial DNA transfer is the newly formed phages now carrying bacterial DNA. When they comes out of the cell, they carry to or they carry bacterial DNA. They can infect recipient bacterium, the next group of bacterium which was not earlierly. Just look at this one. This bacterium was not earlierly affected by the manipulated DNA, but now this manipulated DNA incorporated into bacterial DNA, and this cause again manipulation to the next gene. If more and more manipulation occur, that lead to more mutation, that lead to more change in the structure. Are you getting my point? This is all
Sir,
about the bacterial transduction.
sir, every random fragment of DNA can undergo the recombination.
Again.
Every random fragment of DNA that is produced from the bacteriophage can able to recombinase in the bacterial genome.
Random fragment of DNA.
Yes, sir.
Not I'm getting you properly. Your question is random fragment of DNA.
The picture number 5.
Picture number 5, yes.
There is a random fragment of DNA that is incorporated by the bacteriophage into the bacteria.
Yes, this is the
The blue
manipulated
color
DNA.
and the pink color.
Yes, these are the manipulated DNA. Just look at this picture number 3. Picture number 2 explains about the process of manipulation. That means the bacterial DNA, this pink color structure. Bacteriophage DNA is the pink color structure. Blue color structure is the bacterial DNA. Bacterial DNA replication process is manipulated by the phage DNA. And this is the newly formed or manipulated DNA. When this manipulated DNA, means mutated DNA, again come in contact with the recipient bacterium, which was earlier not infected, non-infected bacterium, they infect the next group by same manner, by manipulation of this structure. Both the things are manipulated, this one and this one also. Clear?
So both are capable to integrate the bacterial DNA.
Both are capable to integrate. One is bacterial DNA, it has the capability. One is phage DNA, it has also capability of manipulation. They can incorporate into the process.
So they have any similar base sequence in their fragment or
That is their own mechanism. Manipulation process is their own mechanism.
yes,
Okay.
yes, sir.
Now this role of plasmid in DNA transfer. So first come to the plasmid. So what is that plasmid? They're small circular extra chromosomal DNA. Why it is called as extra chromosomal DNA? The bacterial DNA, the main DNA, they form the chromosomal structure, the rounded chromosomal structure. But another genetic material is present in bacterial cell cytoplasm, which is not the part of this main chromosome or main DNA. Okay, they exist separately from this bacterial chromosome. They carries extra genes that are not essential for basic survival. That means if you remove this from the bacterial part, they do not cause harm to the bacterium. Okay, but their presence gives bacterium more advantage. That is the benefit of presence of plasmid. Their presence of or absence do not matter for the bacterium, but their presence gives more advantage to the bacterium. That's why it is written in the second paragraph that plasmid often carry genes that provide selective advantage to the bacterium. Okay. A particular characteristic if present in the plasmid, which is not present in the main DNA, like here one mention is bacterial antibiotic resistance. So many characteristics you can incorporate: toxin production, antibiotic resistance, conjugation abilities, metabolism of unusual substances. They are protective nature. They may or may not present in bacterial genome, but if they present in plasmid, they give more advantage to that particular species of bacteria for the process of survivability. Okay. And more property of this plasmid, that many plasmids can be transferred from one bacterium to another through the process of conjugation, which we have discussed earlier. The process of conjugation by using pili, sex pilus, or sometimes through transformation or transduction. Okay. So different types of plasmids are there. These are the structure of plasmid here. This is main DNA. This is plasmid DNA. Different types of plasmids are there basing on their property, basing on different characteristics. The plasmids are fertility plasmids or aplasmids, which carry genes needed for conjugation. We can call it as conjugative plasmids also. Okay, we can take the example of F factor. In the process of conjugation, we have discussed about the F factor, F plus or F minus. Then another category of plasmid is resistant plasmid. Resistant plasmid means they carry genes that help in bacteria survive antibiotics. They develop resistance against the antibiotic. Then cold plasmids, they produce protein called bacteriocin. It's a toxic that kill other harmful bacteria. It's a protective mechanism. Then virulence plasmid, they carry genes that make bacteria more pathogenic. Okay, so these are different kinds of plasmids. Then come to role of transposon in DNA transfer. So before going to that, first of all, we have to discuss about DNA transfer. So what is DNA transfer? Particularly if we are talking about bacteria, the DNA transfer means movement of DNA, movement of genetic material from one bacterium to another bacterium. What happened in horizontal gene transfer? Different processes already we have discussed: transformation, conjugation, and transduction. Okay. Then we'll go for the transposon. So what is that transposon? It's a DNA sequence. Transposons generally, this structure is here. This transposon is a sequence of DNA that can move from one location in the genome to other location. Transposon movement is called transposon movement. So they are sometimes called as jumping genes. Okay. Here it is mentioned that jumping genes, this is the condition before transposon jumping, transposon before jumping. Okay. This transposon binds to the inverted repeats. Then synaptic complex form. Got it? So this is the some key features of this transposon are they're mobile in nature. They can move from one place to another place within the DNA. They carry genes. They often carry important genes like antibiotic resistance genes, disrupt genes. Got it? Different types of transposons are there. It is mentioned here. insertion sequence element, IS element, it is called as IS element, composite transposons, retrotransposons, insertion sequence means or IS element, they are simple type of transposons. They only carry genes needed for the movement, like the genes for transposes, the enzyme that cut and paste the transposer. Only the cut paste function. Okay. Then composite transposon, larger transposons are they are known as composite transposons. They contain extra genes like antibiotic resistance along with insertion sequence at both the ends. Okay. Then retrotransposons generally in eukaryotes, they move by copied into RNA, the reverse transcribed back into DNA before insertion, which is common in plants and animals. So these are various kinds of transposons, particularly these transposons based on the property of cut and paste. Sorry, focus on the property cut and paste. That means the transposon is cut out of its original location and inserted into a new spot. Okay, that is the basic properties of this transposon. Any confusion in this transposon? If any confusion, then you can ask me. Okay, then we'll go for the next slide. This is the end of this part. So many things we have covered from this unit. Okay, tell me if you any confusion from this, from the bacterial genome evolution. Already we have covered bacterial genome evolution. If any confusion regarding this, you can ask me questions. Otherwise, we will go for the next part that is called as speciation. Okay. So now we are going to the next part. So come to the part speciation. Generally, it is considered as one of the important evolutionary process by which new species are formed. Okay, this is known as speciation. How the new species are formed. In the first unit, when we discussed about the natural selection, the concept of natural selection, we discussed about the formation or origin of new species. In Darwin's concept, Darwin explained about 5 important things. That is enormous fertilization, that all organisms try to produce its own progeny in a faster and in a faster manner. Okay. Then there is scarcity of resources for all the organisms that leads to struggle among the organisms. The struggle leads to what? The struggle leads to competition among them. Okay. That competition is for existence. That's why it is known as struggle for existence. And this struggle for existence results survival of certain species. Those species survive, they form, they become the fit. And those fit one, they develop certain properties to adapt themselves in the nature. And this development of certain properties for the survivability of organism is known as natural selection. And those organisms selected by nature, they become the new species which are different from the primitive group. For this concept is known as origin of new species, or we can say it as speciation. So in this chapter, we are going to discuss about the speciation, its category, the mechanism of speciation, how speciation occurs. Okay. So these are different modes of speciation. Here, speciation is defined as the process of through which new species arise from existing one. New species arise from existing one is known as speciation. There are several modes of speciation based on geographical and reproductive isolation mechanism. Allopatric speciation, parapatric speciation, sympatric speciation, okay, peripatric speciation. So these are different modes of speciation. Some diagrammatic presentations are there. We are going to discuss each and every one by one. So basic steps for this speciation is isolation. That means when a population gets separated, either geographically or reproductively, that is one of the important cause for this speciation. Genetic divergence is another cause. Over time, mutations, natural selections, genetic drift, they cause separated groups to become genetically different. And the last important one is the reproductive isolation. If the organism, the newly formed organism, they reproductively isolated from the primitive one, that is also another mechanism for speciation. Okay, so these are different concepts, different steps of speciation. First of all, come to allopatric speciation. Allopatric means this known as geographic speciation. Allopatric means geographic speciation, geographically separated species. This is a type of speciation that occurs when group of the same species become isolated from the rest of the population. And this isolation can be caused by a physical barrier like desert, river, or by great distance. This is the pictorial presentation of this allopatric speciation. This is the original speciation, okay, which has a common gene pool. That means they all are living together. This original population, if separated from each other due to habitat fragmentation, due to geographical barrier. If a river passed through this landmass or in between this landmass, that cause the separation of these two. And this group of species, by the course of time, they completely separated. They develop new adaptability with the changing environment, and they become separated from the primitive group. So they become reproductively isolated from the primitive group, and it results formation of two different groups. Okay, this is the presentation. The group of species, how the course of time they get separated from each other. Okay. So this characteristic of allopatric speciation, allo means other, patric means homeland. How allopatric speciation happens? It's a physical barrier like river, mountain, glacier, desert. They split the original population into two groups. Independent evolution is also another cause. Like each group faces different environments, mutations, natural selection, pressure. Okay, divergence, genetic divergence is also another cause. The cause allopatric speciation, accumulation of differences in DNA, appearance, and behavior. So these all are responsible for this allopatric speciation. Okay, the basic characteristic is are isolation, that means physical barrier, gene flow between the isolated group and rest of the population, evolution over time, isolated group evolve into separate species. Already I have explained in the picture. Now come to the examples of allopatric speciation. The best example is Darwin's finches. That means finches of different Galapagos Island evolved into different species based on food source available. You know that evolution is due to environmental pressure. When all the Darwin's finches are present in the main island, a single type, a common type of food resources available at that island. That's why all the finches, they are looking alike. But when they move to different area having different food resources, that leads to creation of new environmental challenge for those species. To make them survive in the new environment, they do what? They adopt themselves. They try to make certain changes in their own ability, own characteristic to get the food, to develop proper adaptability to the environment. Then only they can survive. If they survive, that means nature selects those species. Darwin's finches, when different islands having different food resources, different seed structure, Darwin's finches develop different structures of beaks. And those beak structure make them reproductively isolated from the original population. There is a geographical barrier, the sea barrier, and when they move to the next island, there the food resources are completely different. They develop their different beak structure from the original population, and at last, after many generations, they found that the new generation is completely different from the original generations. Okay, that is the example of Darwin's finches. for allopatric speciation. I think all of you clear about allopatric speciation. If any confusion about allopatric speciation, you can ask me.
Sir, what is gene flow?
Yes, gene flow. I'm just discussing it in brief. I'm going discuss in the next class the gene flow in detail. The transformation, the movement of genes, the alleles from one population to another population is called as gene flow. When it happens when individuals migrate and reproduce in different populations. Clear?
Yes, sir.
Sir, what are the
Any
importance
confusion?
of allopatric speciation? Importance क्या है?
Importance of allopatric speciation.
Yes, sir.
Importance in evolution. We can say it as in two ways. It is important. The first formation of new species is one of the important, and second one is the diversity, increasing diversity, species diversity.
Okay.
Okay. It also encourages the adaptation process. It is another importance. Got it? Got your answer?
Yes.
Okay. So for this, up to this for today's class. Tomorrow we're going to discuss the next and rest part. Tomorrow, I think the last class for this paper. We are going to discuss the class tomorrow. Okay. Next speciation, human evolution, and all others. One or two more questions.
Sir, I have one question.
Yes.
Sir, can you cite another example for allopatric speciation?
Another example of allopatric speciation is also mentioned here. The separation
Yes,
of
sir.
marine creatures on either side of Central America when Isthmus of Panama closed. So many examples are there for this allopatric speciation.
Sir, two example of prokaryotes and eukaryotes.
Two examples of prokaryotes and eukaryotes. So many examples I have cited. Bacteria is one of the example of prokaryote. Animal cells, eukaryotic cells, plant cells, eukaryotic cells. Okay.
Okay, thank you, sir.
Okay. Then good night to all.
Thank you, sir.
सर पीपीटी पठे
Thank
ना
you,
दिखे
sir.
PPT पर ग्रुप पे सेंड ହେଇଛି
ग्रुप सेंड है
ग्रुप
नहीं
पे ना होगी
हाँ
ग्रुप रे
ग्रुप रे
PPT
अच्छी
ଯାଇନି
ना सर
ଗ୍ରୁପ ରେ ଅଛି
ତାହେଲେ ତୁମେ
Okay,
ପିପିଟି
ତୁମ
starting
ନମ୍ବର ଟା ଦେଇଦିଅ ସାର୍ ତୁମକୁ ସେଣ୍ଡ କରିଦେବେ।
सर को मैसेज करছি सर
ନାଇଁ ଶୁଣ ମତେ ଇଣ୍ଡିଭିଜୁଆଲି ଏତେ ଲୋକଙ୍କୁ ପଠେଇବାଟା ଡିଫିକଲ୍ଟ ହଉଛି
ସାର୍ ସାର୍ ଆପଣଙ୍କୁ
ସମସ୍ତେ
ଗ୍ରୁପ ରେ ଆଡ୍
individually କହିଲେ
କରିଦେବେ।
ମୁଁ କେତେ ଜଣଙ୍କୁ ସେଣ୍ଡ କରୁଥିବି? ନାଇଁ ଆମେ ତ ଟିଚର ମାନେ ଅଲଗା କିଏ ଗ୍ରୁପ ରେ ଅଛନ୍ତି କି? ଟିଚର ତ କେହି ନଥିବେ ତମ ଷ୍ଟୁଡେଣ୍ଟ ଗ୍ରୁପ ରେ।
କେବଳ କୋଅର୍ଡିନେଟର ସାର୍ ଅଛନ୍ତି।
କୋଅର୍ଡିନେଟର ସାର୍ ଥିବେ, ଅଲଗା କେହି ଟିଚର ନଥିବେ।
ନା ଟିଚର କେହି ନାହାନ୍ତି।
ନାହାନ୍ତି,
ଠିକ
ବଟ୍
ଅଛି
ଜଣେ
ତମର
ସାର୍
ଯଦି
କହୁଥିଲେ ଆମକୁ ବି ଆଡ୍ କରିଦିଅନ୍ତୁ ଗ୍ରୁପ ରେ ବୋଲି।
ନାଇଁ ମତେ ଆଡ କରିବା ଦରକାର ନାହିଁ। ତମର ଯଦି କେବଳ ଜଣଙ୍କର ପ୍ରୋବ୍ଲେମ ଅଛି ତମ ଜଣକୁ ହିଁ ମୁଁ ଆଉଥରେ ସେଣ୍ଡ କରିବି, ନହେଲେ ଆଉ ସେକେଣ୍ଡ ଇଏ ଆସିଲେ ମୁଁ ସେଣ୍ଡ କରିପାରିବିନି। ତମ ଭିତରେ ହିଁ କୋଅର୍ଡିନେସନ କରିକି ତମେ ସେଣ୍ଡ କରିବ। କାରଣ ସମସ୍ତେ କଲା ବେଳକୁ ମେସେଜ ବହୁତ ଗୁଡେ ମେସେଜ ଇଣ୍ଡିଭିଜୁଆଲ ମୋ
ହଁ
ପାଖକୁ
ଠିକ ଅଛି
ଆସୁଛି।
ସାର୍। ଓକେ ଓକେ ସାର୍।
ଠିକ ଅଛି, ଓକେ।
ଠିକ ଅଛି ସାର୍।
Okay, thanks to all for your patience.
Yes, sir.